So I'm Yolanda. I'm a postdoc in Adam Arkin's lab at Lawrence Berkeley National Lab. And I'm excited to tell you about my main postdoc project on using barcoded expression libraries to study gene functions in the gut among gut bacteria. So over the past few decades, we've seen an explosion in the number of studies on the human gut microbiome. We've come to appreciate that trillions of microbial cells live within and on the human body, and that microbial genes actually outnumber human genes at about 150-fold. And in particular, the human gut is one of the most densely populated microbial habitats on Earth, and we're only beginning to understand how gut microbes contribute to our health and disease states at a mechanistic level. So despite the large amounts of sequencing and omics data we have on the gut microbiome, identifying causation between genotypes and phenotypes is still quite difficult. And this is because bacteria are extremely diverse and most genes have unknown functions. So specifically in human stomatic genomes, we can see that most genes are annotated as hypothetical proteins or are only vaguely annotated. To tackle this problem in the microbiome field, functional genomics can be used to rapidly connect genes to phenotypes, which can uncover novel gene functions. For instance, our lab and collaborators have applied DNA barcoding to develop loss of function, RBTNC libraries. This stands for randomly barcoded TNC libraries. So this allows us to quantify individual mutants within a pooled library, and this can drastically increase the throughput use of each library across many conditions. And today, RBTNC libraries have been generated for over 40 isolates and tested in competitive fitness assays across hundreds of conditions. You can see all the data on the fitness browser. So this approach really allows us to interrogate gene functions at scale, but loss of function libraries are restricted to single genomes and also limited to genetically tractable isolates. So for my postdoc, I wanted to build a similar platform, but for expression libraries. So gain of function libraries, which can capture genes from multiple genomes and also uncultivated isolates by using metagenomic DNA. So I'm excited to present my new workflow, which we termed BOBA-seq, where barcoded overexpression bacterial shotgun library sequencing. And here we leveraged advances in long-read sequencing to map barcodes to insert fragments. We also generated an empty vector library that is highly diverse with about 14 million barcodes. And we also use a gut anaerobe theta as the expression string. So B theta is from the order Bacteroides, which are highly abundant in the human gut, and they're known for metabolism of complex carbohydrates. But more importantly, they're also fast growers and they are genetically tractable, so serving as a suitable expression platform for gain-of-function libraries. So for the initial study, I constructed 7 libraries using genomic DNA from Bacteroides and cloned DNA fragments into barcoded vectors. So these are randomly shared DNA fragments. Using barcode sequencing, or BARseq, we determined that each library was about 40 to 100,000 in barcode diversity. And then using PacBio sequencing, we map barcodes to inserts for each library. So most of the genes on each genome are completely covered by at least one fragment in our libraries. And the individual libraries are conjugated into B theta and then pooled. So for the initial fitness screen, I pooled 6 of these libraries and screened, which consisted of 300,000 strains encoding about 21,000 protein coding genes. And this pooled library was then assayed across 53 conditions with the inducer. This consisted of 41 single carbon sources, and that includes complex carbohydrates as well as simple carbohydrates. And I also tested 5 inhibitory compounds at a couple of different concentrations. So growth was observed in 41 conditions, and these were harvested for barcode sequencing. So from the barcode sequencing data, we then calculate a fitness score for each barcode. And this is essentially the log2 ratio of the barcode abundance after growth in the selective condition over time 0, where the pooled library inoculum. And here, because this is a gain-of-function screen, we're looking for positive, strong positive fitness scores. So these are strains that contain genes that confer the benefit in a specific condition. So this is showing all of the bar-seq data or fitness scores from the 41 conditions. And we see a strong correlation between the two replicates. We identified over 300 high confidence positive scores in both replicates. And this led to 26 unique beneficial regions that were enriched across 15 conditions. And at 40% amino identity, this led us to 13 protein clusters. And more excitingly, we observed new phenotypes for nine genes. And this includes genes expected to confer function based on known functions, to confer benefit based on known functions or annotation, as well as genes that were previously uncharacterized and less expected. So here I'll just go over a couple examples for each of the two categories. So in terms of genes that are consistent with known functions or annotations, we observed the enrichment of dihydrofolate reductase in trimethylpring. And this is the enzyme target of trimethylpring antibiotics, so that made a lot of sense. We also observed the enrichment of two kinases in D-glucosamine. So these enzymes are likely phosphorylating glucosamine or other intermediate in the pathway for carbon utilization of this metabolite. So here I'm showing fitness score versus fragment plots. On the y-axis, we have fitness scores. The x-axis is the genomic position of the genome of that library. And highlighted in green are genomic fragments that completely cover this gene that we think is causative for the gain in fitness. So you can see that strains containing fragments in this region led to increase in fitness scores. Encouragingly, some of our hits are also supported by complementary fitness data from the B-theta, RBT, and seq loss of function library. And this provides additional evidence for our gain-of-function data. And in terms of previously uncharacterized genes or more novel functions, we also found three TOMB-dependent transporters that seems to be involved in raffinose uptake. One of these transporters also conferred a benefit on fructooligosaccharides. To our knowledge, TOMB-dependent transporters have not been reported to uptake raffinose, so this is quite exciting. And the other region that was pretty novel is a region that encodes a single-stranded DNA binding protein. And this region conferred a fitness gain on fucose. We were also able to confirm that this region conferred a growth advantage using single strains. So these are three fragments that we observe in our fitness data compared to the empty vector control. So we're currently working to characterize this hit in more detail. So with this, I will briefly summarize our work. That BobaSeq is a pretty powerful platform. It enables fitness assays of DNA barcoded expression libraries to uncover novel phenotypes for an important group of gut anaerobes. I think this approach will lead to many interesting results in the future as we scale up to additional libraries as well as new conditions. And the second note I'll leave you with is that this workflow can be applied to study gene functions in other bacteria or ecosystems. So all you need are vectors and a suitable expression string. I would be happy to collaborate and help with your project. So feel free to reach out. And with this, I would like to thank my PI, Adam Arkin Lab, as well as co-authors on the paper. I will add a note that the Arkin Lab and Metallic Lab are jointly looking for postdocs to work on bacteriophage interactions. So if you're interested, definitely reach out.